DNA is short for deoxyribonucleic acid. It is made up of multiple nucleotides. A nucleotide consists of a nitrogenous base, a ribose sugar, or deoxyribose sugar, and a phosphate group. First of all, not all nitrogenous bases are the same. In DNA, there is adenine, guanine, thymine, and cytosine. We symbolize these different types using the first letter of each type capitalized. Adenine and thymine are paired, and guanine and cytosine are paired. This is known as complementary base pairing. Hydrogen bonds connect the complementary bases. The hydrogen bonds are symbolized using the magnets. Next, for the sugar pentose, there are 5 carbons and 1 oxygen, which is on one of the corners of the pentagon. The oxygen is symbolized by the purple. For the phosphate group, it is symbolized by the letter P attached to the sugar. All of the bonds within a nucleotide are covalent bonds because the electrons are being shared. The complementary base pairing allows for 2 strands of chained nucleotides. These strands are antiparallel, where the oxygens on the sugar pentose face the opposite directions. For DNA replication to occur, the helicase enzyme unzips the complementary base pairing by breaking the hydrogen bonds between them. Now that the strands are unzipped, free nucleotides bond with hydrogen bonds to these strands by the DNA polymerase enzyme. This action is represented by the hand pairing the bases together. These newly bonded nucleotides are called new strands, while the original strand is known as the parent strand. DNA replication is a semi-conservative process because half of the existing DNA strand, the parent strand, is always conserved. To make proteins, 2 sets of reactions are needed to take place: transcription and translation. Transcription starts by the unzipping of DNA strands by the RNA polymerase enzyme. Then messenger RNA is formed by pairing to the antisense strand, which is one side of the DNA. Notice that there is no thymine base for mRNA, but instead there is uracil. Also, the sugar pentose in mRNA has a ribose, different from the deoxyribose in DNA. Translation starts with the mRNA venturing out of the nucleus to find a tRNA in the ribosome. The 3 bases on the middle loop are called anticodon bases. The triplet mRNA codons will pair with the 3 anticodons from the tRNA. Several of these amino acids will group together to form a polypeptide. A polypeptide is considered a protein when it can carry out a function. Sometimes polypeptides can only function when they combine with other polypeptides.
Thanks for watching!